In this lecture, we are going to learn about enzymes. As you know, enzymes are proteins. They are synthesized by biological cells and in all organisms. Their most important function is they have powerful catalytic activity. They catalyze several reactions in body and also in food industry. Enzymes are extracted from edible plants and the tissues of food, animals, as well as those produced by microorganisms such as bacteria, yeast, and fungi. Most enzymes used today in the food industry obtained from microorganisms. So, enzyme catalyzed reactions proceed in many foods. Those reactions can enhance or deteriorate food quality. Examples for enzyme catalyzed reactions in foods can be such as ripening of fruits and vegetables, the aging of meat and dairy products. Also, the processing steps involved in the making of dough from wheat flour involves enzymes too. Several enzyme classifications can be done. If we want to make a classification based on source, enzymes can be generally classified in two ways by source, food enzymes. There are enzymes from exogenous sources that exist in foods and which may or may not be responsible for reactions that affect food quality. Endogenous enzymes. So they exist in food. In food matrix. Okay, they are in the food like this, edible plants. Edible plants are endogenous sources. Okay, apple has it. There are also enzymes from exogenous sources. So these are added to food products to cause a desirable change in food. And they can be obtained extracted from a variety of sources. Such as you add an enzyme to tenderize meat tenderizer enzyme. So you are adding these exogenous enzymes to food products to cause a desirable change. The functionality of these enzymes depends on many factors. Appropriate functionality of enzymes relates to the catalytic activity, selectivity, and stability under the conditions during a specific application. When selecting an enzyme, wide temperature and pH range conditions are desirable. Okay, let's continue. We said enzymes are proteins. Some enzymes consist only of proteins. Many enzymes consist of a protein and a non-protein part. So non-protein portion is called cofactor. Non-protein part. So for some enzymes, this cofactor is necessary for the enzyme's catalytic activity. Here this protein part is inactive. Okay, and this part is active. Activity comes from the cofactor. So when an enzyme requires a cofactor for its activity, okay, the inactive protein component, which is referred as apoenzyme, and this apoenzyme plus the cofactor is called holoenzyme. Okay, so in first situation we have active proteins, the enzyme is active, enzyme has catalytic activity. In the second situation we have inactive protein part, and we need a cofactor for enzyme's activity. So what is this cofactor? A cofactor may be another organic molecule. So if it's an organic molecule, in this case it's called a coenzyme. Usually coenzymes are vitamins or made from vitamins. Okay. Or cofactor can be inorganic molecule. Such as metal ion. Okay. These metal ions can be iron, manganese, cobalt, copper, zinc. Okay. Maybe zinc, even calcium, etc. Okay, minerals. So we always need vitamins and minerals in our diet. Now, maybe you will understand that the need for minerals and vitamins in the human diet is partly attributed to their roles within metabolism as cofactors and as coenzymes. Okay. Just I want to clarify one point here with apoenzyme. We said this is inactive. The apoenzyme is inactive, and it needs cofactor to be active as an enzyme. But if that part, apoenzyme, denatures, again, this enzyme loses its catalytic activity. It won't be active anymore if there's a denaturation of that protein part, apoenzyme. Okay, let's move on. So, specificity of enzymes. Enzymes are often very specific in the type of reaction they catalyze. So different enzymes catalyze different reactions. They are specific. Like we have absolute specificity, relative, or stereochemical specificity. Let's look at the absolute specificity. So an enzyme with absolute specificity catalyzes the reaction of one and only one substance. Here we have an example, glucokinase. So this enzyme glucokinase catalyzes the only reactions of glucose. It acts on glucose only. Glucokinase. So this enzyme has an absolute specificity. Okay. Okay. So an enzyme with relative specificity catalyzes the reaction of structurally related substances. A group, a bond linkage, for example, lipase and protease. So remember, lipase hydrolyze lipids. Protease is an enzyme for proteins. So as an example, split up proteins. Okay. So there are also enzymes with stereochemical specificity. So this catalyzes the reaction of only one of the two possible stereoisomers. Remember, we have L amino acid and D amino acids. As an example, here we have L amino acid oxidase enzyme. And this enzyme catalyzes the reaction of L amino acid oxidation, but not D amino acids. Only L form. Okay? Enzymes are specific in the type of reaction they catalyze. Okay, let's see how enzymes work. So enzymes have an active site. So this is part of the molecule that has just the right shape and functional groups to bind to substrate. It's important. It should be right shape. And there are also functional groups on this active site. Shape is important. The substrate molecules bind to active site of enzyme and they undergo a chemical reaction. Active site is a small region on the surface of the enzyme. Here we have active site. If this is an enzyme, active site only here. Or in another model, only here. It's a small region. But there are many functional groups, so chemical reactions can occur. So when the substrate binds to the active site via some combination of intermolecular forces, an enzyme substrate complex is formed, like this or like this. So the chemical transformation of the substrate occurs at the active site. Okay? The functional groups on the active site participate in the making and or breaking of chemical bonds. So active site typically binds substrate reversibly. It's a reversible process. It binds via non-covalent forces. Okay. Substrate, as for substrates. Reversibly, it's important. Via non-covalent forces. So after the conversion is complete, after the reaction is complete, the product is released from the active site, leaving the enzyme free to react with another substrate molecule. So there are two hypotheses regarding how an enzyme works. We have the first one is key and lock, or lock and key hypothesis. And the second one is induced fit theory. So the key and lock theory explains the high specificity of enzyme activity. It's the simplest model to represent how an enzyme works. The substrate simply fits into the active site to form an enzyme substrate complex. Okay, high specificity. So specific shapes and size are important. I will just say S. So enzyme surfaces accommodate substrates having specific shapes and size. So only specific substances fit in an active site to form an enzyme substrate complex. Okay? So there is a limitation of this key and lock theory. So this theory requires enzyme conformations, enzyme shapes to be rigid here. However, research suggests that the studies suggest that instead enzymes are at least somewhat flexible. Enzymes should be flexible. That's the limitation of this key and lock theory. Therefore, this theory needs a modification. So a modification of this key and lock theory called the induced fit theory. So induced fit theory proposes that enzymes have flexible conformations. The shapes, the conformation is flexible. And they may adapt to incoming substrates. Okay, induced fit. Enzymes adapt to incoming substrate. Okay, in this model, the enzyme molecule changes shape as the substrate molecules gets close. The change in shape is induced by the approaching substrate molecule. That's why it's called induced fit. Induced by the approaching substrate molecule. The active site adopts a shape that is complementary to the substrate. Okay, this is more sophisticated model theory. It relies on the fact that enzymes are flexible. Okay, let's move on. Enzyme catalyzed reactions. So let's consider the catalysis of a chemical reaction. So the first graph belongs to an uncatalyzed reaction. And this is enzyme catalyzed reaction. So this is enzyme and this is substrate. So in an uncatalyzed reactions, there are reactants. They just randomly bump into each other. They collide randomly and react randomly. So we have substrate and there is an activation energy required to reach the activated transition state. In both cases, we have transition state. So at this transition stage, chemical bonds are formed or cleaved in order to yield some intermediate products, which is then released as product at the end. So this chemical reaction can occur without an enzyme, and we can convert the substrate to product. However, if we use enzyme, we will accelerate the reaction rate. So the same reaction can be accelerated by the addition of a suitable enzyme. So enzyme transforms reactant S into some intermediate products like ES, EP. Then at the end forms product. So as shown here, the transition states of enzyme catalyzed reaction are at a lower energy level than the transition state of an uncatalyzed reaction. So this is the transition state of enzyme catalyzed reaction. The level is here. And this is a transition state for uncatalyzed. So what does that mean? That means enzymes lower the activation energy of the reaction. So it accelerates the reaction. So without enzymes, most cellular reactions in the body would take place too slowly to support life. Enzymes can also accomplish in seconds what might take hours or weeks under laboratory conditions. So what happens to enzymes after reaction? So enzymes participate in the reaction, but they remain unchanged at the end of reaction. So they increase the rate of chemical reaction without being used up in the process. So they may be used over and over. They are still active. So they have activity to catalyze other reactions. Okay? So enzymes can only change the rate of reaction, not the position of the equilibrium, okay? It reached the same equilibrium, the same position, and at the end we have the same product. It's just an enzyme catalyzed reaction takes a different route, okay? As you see here, different route. So at the end we have the same product. The position of the equilibrium is the same. Okay, let's continue. So enzymes are extremely efficient catalysts. For example, here there's a comparison with an acid catalyst. We have invertase enzyme. It catalyzes the hydrolysis of sucrose molecule. Then it forms glucose and fructose as products. And here you see the relative reaction rates. If we use acid catalyst in this reaction, it's just 1. But when we use enzyme, the reaction rate dramatically increase, like 10 to 9. So enzymes are extremely efficient catalysts. Okay, let's continue. So enzyme nomenclature. Enzymes typically have common names, often called trivial names. So these refer to the reaction that they catalyze with the suffix ase. As you see here, ase, such as peptidase or oxidase, dehydrogenase. So we have suffix ase, ase. Yeah, sometimes they also indicate the substrate on which the enzyme acts, such as glucose oxidase. So we have a table here, and so here we can predict the general enzyme names and the reactions they catalyze. For example, we have decarboxylase. So it catalyzes the removal of carboxyl groups from compounds. So here, carboxyl groups. We have phosphatase. So here, it catalyzes the hydrolysis of phosphate ester linkages. So peptidase, hydrolysis of peptide linkages. Esterase, formation of ester linkages. So all they are related. Okay. So in some cases, suffix in also used. It's some of the earliest enzymes to be discovered were given names ending in. The reason for that, because enzymes are protein. So to indicate that enzymes are proteins, they were just adding in. For example, so today still we call it that way. Some digestive enzymes, we have pepsin, trypsin, or chymotrypsin. Okay? Because they were proteins, that's why. So after that, there are like large groups of enzymes are discovered. So therefore, due to this growing complexity and inconsistency in the naming of enzymes, the International Union of Biochemistry set up the Enzyme Commission, AC, Enzyme Commission classes. So to address this issue. So according to that, enzymes are grouped into 6 major classes on the basis of the reaction they catalyze. So all EC names end in ase. Okay? We have oxidoreductases. They catalyze the oxidation and reduction reactions. Okay? So they are related like hydrolases. Catalyze the hydrolysis of chemical bonds. So it goes like that. We have 6 major classes. It's on the basis of the reaction that they catalyze. Okay, let's continue. So how do we express the enzyme activity? So enzyme assays are experiments that are performed to measure enzyme activity. So terminology related to the rate at which an enzyme catalyzes a reaction has been there's a standardized. So the general unit of enzyme activity is U. Okay. So U is defined as that which causes the conversion of 1 micromole substrate per minute under optimized conditions. So enzyme activity refers to the catalytic ability of an enzyme to increase the rate of a reaction. So the molecular activity of enzyme is defined as the turnover number. It's the number of substrate molecules that can be converted by one enzyme molecule per minute under defined conditions. So it's the number of molecules of substrate acted on by one molecule of the enzyme per unit. So per minute. So enzyme activity is about the quantity, okay? So when we talk about turnover number, it's about the conversion of substrate molecules, okay? So if you buy an enzyme, you will see the units of activity on the bottle. Okay. So let's learn what factors affect the enzyme activity. So there are several factors affecting catalytic activity of enzymes. Various environmental factors are able to affect the rate of enzyme catalyzed reactions. It can be like reversible or irreversible changes in protein structures. So the proteins in enzymes are usually globular proteins, enzyme proteins. They are globular. Remember, we have globular and flexible proteins. Generally, enzyme proteins are globular. So the inter and intermolecular bonds that hold proteins in their secondary and tertiary structures. And these are disrupted by changes in pH, temperature, pressure, water activity, substrate concentration. There are many factors. So let's learn the most important ones. So presence of water affects enzyme activity in several ways. It can be a diffusion medium, or it can act as a co-substrate, or it can stabilize protein. So enzyme require a certain level of water in their structures, so in order to maintain their natural shape, natural conformation, which allows them to deliver their full functionality. So how does water affect enzyme activity? So regardless of the type of reaction, the functionality of enzyme itself is maximum at an optimum level of water. Okay, we are talking about an optimum level of water. So here you see enzymes like amylases, lipases. So different enzymes have a different optimum level of water for their activity. So beyond that optimum level of water, the enzyme performance is declined due to the loss in enzyme stability. So as you see in this graph, some enzymes exhibit maximum activity about like 0.9 water activity. But some enzymes can remain active at very low water activity. Here, for example, lipases. So the optimum level of water changes depending on the enzyme. So this low water activity, so given the length of time some food products are stored, So this can become significant. Even with very little water, there could be some deteriorative enzyme activations causing some like off flavors, so which is undesirable. So at this very little water activity, lipases catalyzes the maybe hydrolysis of 3-acylglycerols and forming some free fatty acids, which leads to off flavors, formation of off flavors. So freezing affects the enzyme activity, freezing of the water present. So fast freezing creates smaller, more dispersed pools of unfrozen liquids. So enzyme activity is diminished with fast freezing. Decrease. So slow freezing creates larger pools of unfrozen liquid where enzyme and substrate can interact. And also when we look at the melting, slow thawing is very detrimental to enzyme activity. Because it promotes frost formation or ice recrystallization during thawing, and it's detrimental to enzyme activity. Okay. Okay, let's continue. Enzymes are also sensitive to pH change. So changing the pH of its surrounding will also change the shape of the active site of an enzyme. This contributes to the folding of the enzyme molecule, its shape and the shape of the active site. So changing the pH will affect the charges on the amino acid molecules on the active site. Okay, the pH affects the state of dissociation of functional groups involved in the enzyme substrate complex. So enzymes have an optimum pH, and extreme pH values cause enzyme denaturation. Okay? There is an optimum pH for an optimum activity. So each enzyme works within a quite a small pH range. So the pH range over which an enzyme is stable is usually larger than pH range for enzyme activity. So stability in bar graph at the right measured at typical food processing conditions. So activity and stability. So they are different. Let's continue with temperature effect. So how temperature affects the enzyme activity? As the temperature rises, reacting molecules have more and more kinetic energy. So this increases the chances of a successful collision. So the reaction rate increases. So there's a certain temperature at which an enzyme's catalytic activity is at its greatest, like this. So this optimum temperature is usually around human body temperature, like 37.5 degrees Celsius. It's for enzymes in human cell. So above this temperature, the enzyme structure begins to break down, denaturation. You know, at high temperatures, intra and intermolecular bonds are broken. Okay, above or below the optimum temperature, the rate is lower. The reaction rate is lower. Okay, there are two phenomena. First one, as temperature increases, free energy of the system increases, lowering the energy barrier for reactions to occur, and reaction rate increases. The second one, as temperature increases, enzyme stability decreases because of the denaturation. Okay, let's continue. So enzyme and substrate concentrations affect enzyme activity. The concentration of enzyme is typically low compared to that of the substrate. So increasing enzyme concentration also increases the rate of the reaction. So here. So the rate of the reaction is directly proportional to the concentration of the enzyme. So it's the linear straight line. So that means doubling enzyme concentration doubles the rate of the reaction. So graph of reaction rate versus enzyme concentration is a straight line. So on the other hand, the concentration of substrates also affects the rate of reaction. So here, increasing substrate concentration increases the rate of reaction. But eventually the rate reaches a maximum level, Vmax, and remains constant after that. So the maximum rate is reached when the enzyme is saturated with substrate and cannot react any faster under those conditions. Okay, they were the important factors affecting enzyme activity. That's all for this lecture. Let me know if you have any questions.